Thank you everybody for logging in and coming to Suni Shah Walter's seminar here. I've known Suni for quite a few years, and I'm really looking forward to this presentation. We kind of intersected at Woods Hole Oceanographic Institution when she started working with some of the scientists there, I guess for her first postdoc, which was at the Naval Research Laboratory when I was finishing my postdoc. And then she ended up getting a second postdoc at the Woods Hole Oceanographic Institute. And where we intersected was in the laboratory facility that measures radiocarbon. So before that, Suni graduated from Columbia in a chemistry department and was really sort of didn't want to follow a lot of her contemporaries into the pharmaceutical industry, but really didn't have an idea that ocean sciences required some sort of a chemistry background. So at that point, she did an internship at the USGS, and they had a project available which involved babysitting and nurturing a finicky instrument, chemical analysis instrument that they figured, hey, you know, a chemistry undergrad would be great for this. And for a year, Suni did that work and kept things running, and probably knowing her, even got it running better than it was before. And then went on to do a master's at the University of Michigan and finally a PhD at Harvard. We fast forward through those postdocs, and she found herself interviewing at the University of Delaware, where she is now at the Lewes campus, which is the sort of marine campus of the University of Delaware. And it turns out that on the chair of the hiring committee, or somebody on the hiring committee, was a person that was a postdoc at the same time that she was doing her internship at the Woods Hole USGS. They were a postdoc there and had known each other. So a little bit of serendipity mixed in there, as Suni puts it, but I'm sure that the hiring was more on merit than serendipity. But in any case, Suni's going to be telling us today about some of the work that she's doing that merges organic carbon cycling and microbial metabolic processes in cool crustal fluids. So I'll hand it over to you, Suni. Thank you for agreeing to a seminar.
Thank you for inviting me. I'm really glad to have been able to come here, and it was a really nice meeting that I had with the grad students earlier. That was longer than I expected it to be, actually, but that was because it was so good. So when I gave Brad that story, I actually kind of put the emphasis on the fact that it was that I was good at running instruments, that I think that gave me those sort of opportunities to go to the USGS. And I was also going to say that because I'm talking about hydrothermal fluids, I wanted to point out that the only reason that I got involved in this whole field of hydrothermal vent and hydrothermal fluid research is that I was also good at taking apart and putting back together mass specs. So if you're, one of those students who's wondering whether it's better to just get your measurements done and get your samples run, or whether it's worthwhile to kind of dig in and become a real expert in your instrument, becoming a real expert in your instrument has served me really well. Okay, so what I was going to talk to you today about is sort of the organic carbon biogeochemistry of hydrothermal fluids that are cooler than what you might normally think about. So I've illustrated that in this cartoon by these blue dots and these purple dots. So down below the water column and also down below the sediments, you have the basalt crust, the oceanic crust, and the crust is more permeable than the sediments, and you have fluids, seawater and altered seawater that's flowing through the crust, warming up, absorbing lithospheric heat, and when it has an opportunity and it's buoyant enough, discharging back into the deep ocean. And we don't know very much about the sort of biogeochemistry of these fluids. So when I say hydrothermal fluids, hang on a second. My slide advancing is different than I thought it was going to be. Oh, I can't advance the slide. Hang on, there we go. Sorry about that. Okay, so when I say hydrothermal fluids, you probably think of fluids that look like this, right? So high temperature, focused flow, that can be a few hundred degrees, or cooler flow that is represented by these sort of shimmering fluids that you see sort of on the side of these kinds of vents, cool enough to support chemosynthetic microbes, and they support these large biological communities that are based on the sort of chemosynthetic, autotrophic bacteria. There's strong contrast, strong temperature contrast and geochemical contrast between the fluids that are coming out of vents like this and bottom seawater. So it turns out that these are actually not very hard to find. They were first found in the 1970s based on temperature anomalies in the water column, and many more vents have been found since. The fluids are acidic, they're anoxic, they're reducing, and they have things like high concentrations of hydrogen, methane, ammonium, iron, and low concentrations of things that you would find in seawater, like magnesium. The magnesium gets stripped away. So all of these kinds of vents are found in locations where you have seafloor spreading. In general, they're found on oceanic ridges where you have magma pretty close to the surface and this intense heating that sets up the circulation pattern where you have seawater that's kind of infiltrating the crust farther on the flank, and it sort of undergoes this high temperature water rock reactions, and warms up and gets buoyant and comes out through the crust in these vents. This happens, like I said, on oceanic ridges where there is seafloor spreading most often. And during this process of seawater entrainment and reactions and reintroduction into the deep ocean, there's also implications for the carbon cycle. The organic carbon that's in the seawater can get decomposed abiotically into CO2 and methane. You can also have the addition of magmatic carbon, and you can tell this from the isotopic composition of that carbon. There can be abiotic synthesis of organic carbon. There can also be biological synthesis for the microbes that are living off of the reducing compounds in these vents. But in general, we don't think of this kind of environment as important for the carbon cycle because they're limited to just a narrow space. So if you're looking for vents, you find them kind of in these along ridges pretty much. So here's a map of all of the known vents in red and the hypothesized vents in yellow. And if you sort of look at this map and then look at the figure on the right-hand side, this figure, here's the age of the ocean crust. All of the hydrothermal vents that you see, pretty much all of them that you see, are in these really red plate boundary areas where there is seafloor spreading. So you're seeing these hydrothermal vents on really young crust where new crust is being created. So the question, or well, I guess the thing that I'm going to try to convince you of now is that if you just look at hydrothermal fluid circulation and hydrothermal fluid biogeochemistry along these red areas where there's new crust being created, you're missing a lot of the global picture, the big picture of hydrothermal fluid biogeochemistry. Like we have these charismatic vents that capture your imagination right on ridges, and there's a lot of work that's been done to understand the geochemistry and the microbiology of these vents. But just looking at these vents misses a whole lot of other hydrothermal circulation. So to illustrate that, here is a figure that shows hydrothermal fluid flux, like hydrothermal volume fluid flux, from plotted against crustal age. And the space that I have outlined in green goes from 0 million years to 65 million years. So what this shows, the hydrothermal fluid flux, that data is created from heat flow measurements. So you can imagine if you have a slab of ocean crust that's cooling and maybe sediments above it, but then the deep ocean and the cold seawater right above it, you have conductive cooling of the hot crust. And if you go out and actually make some heat flow measurements in many places, especially on young ocean crust, you'll find that the heat flow coming from that crust is lower than you might expect from that conductive cooling model. And that represents the advection of fluids of seawater into the crust. So it kind of acts like a radiator, right? You have cold seawater that is recharging into the crust. It's absorbing lithospheric heat, and then it's coming out and delivering that heat back to the deep ocean. So if you go out and make those measurements, you find that you can see this missing expected heat flux that can be attributed to advective heat loss on crust that is as old as 65 million years. So if you go back and look, the reason that I've made this green is if you go back and look at this plot that has the age of the ocean crust, and you look at all of the crust that goes from the red, zero age crust out to 65 million years where it's green, that represents a large fraction of oceanic crust, just kind of overall. Right, so this is a huge areal extent, and then if you're going to try to turn this area into a volume using porosity and thickness of the crust, you end up with a global pore volume, a global volume of fluid that is approximately 2% of the total ocean volume. And if that's a hard number to relate to, at least it was for me, it's also equivalent to about 40% of the volume of the Arctic Ocean. So it's a lot of fluid that's in the crust. So the volume, I think, is kind of like a cool gee whiz number, but what we would really care about is the flux. So you use these sort of fluid flux measurements, and you extrapolate it out, and what you find is that the total hydrothermal fluid flux, so the fluids that are passing through the crust and then delivered back to the deep ocean from below, that's equivalent to about 18% of riverine flux to the coastal ocean. So this is hopefully I'm convincing you, this is a big flux of hydrothermal fluids, and we don't know very much about what's going on in them. So the other point to make is that if you look at this figure, you can see that in really young crust, the majority of fluid flux happens in really young crust. But young crust, this peak right here goes all the way out to 10 million years. So if you put your boundary at 5 million years, approximately 90% of total global fluid flux occurs on crust that's from 5 million years to 65 million years, and just 10% of it on this, what is probably equivalent to this very red, the most red areas on that plot. Okay, so if we're interested in what happens on the flanks of ridges, the best example that we have, the best ridge flank setting that has been characterized is the Juan de Fuca Ridge flank. So this is the eastern flank of the Juan de Fuca Ridge. It's conveniently accessible. You can fly to Seattle or Astoria, Oregon, and hop on a ship and be there, and with really short transit time, so that makes it advantageous. When you go out there, what you see are rocky outcroppings, and there is sort of a visual cue that there are chemically distinct and reducing fluids coming up out of those rocky outcroppings, right? This is a picture of a microbial mat that's on one of these rocky outcrops. There are lots of these outcrops. They've been named very creatively. They're called things like mama bear, papa bear, baby bear. There's a grizzly bear, and I think there are additional ones like, is it a bear and was a bear? So these, what is understood about fluid flow is that there is seawater that recharges or is entrained into the crust through the rocky outcrops. So sediments are relatively impermeable relative to the basalt that's outcropping above the sediment. So in this location, because it's been so well characterized, we know that it's about 50 kilometers from one outcropping to another one where there's discharge. So one outcropping where there is fluids sort of going into the crust. Those fluids pass through the crust under the sediments. They are undergoing water rock reactions. It's losing its oxygen, it's heating up, and by the time it is buoyant enough to come out from the second outcropping, the chemical composition of the fluids looks very different. They're warmer, they're 25 to 60 degrees. In some locations, they're more like 65 degrees C. And this is a pretty substantial fluid flux, right? It's 5 to 20 liters per second of fluid that's coming out of these rocky outcroppings. The fluids themselves are warm, they're anoxic, they're depleted in magnesium. So one of the things that happens during these water rock reactions is that the magnesium in seawater gets stripped out, and hydrothermal and fluid end members tend to have no magnesium in them, zero magnesium concentrations. So the fluids that are coming out here have very low magnesium concentrations. They're enriched in methane and hydrogen and in ammonium, among other things, also in DOC. But the thing that distinguishes this location from other sort of on-axis hydrothermal vent sites is that the pH is near neutral. So there are some differences, but overall, this kind of looks a lot like regular hydrothermal vent sites, right? Okay, so what if we were really looking to find a site that was representative of that 90% of circulation that happens on older crust? So that's a lot harder to do, because if you're getting outflowing fluids that look not very different, you know, they're cooler, there's not that temperature contrast, they're harder to find. So you can go create a site, and that was what was done. So this is work that was done, a lot of work that was done by Katrina Edwards and Wolfgang Bach many years before I became involved in this project. With the IODP, they drilled into older crust. So this is a site on the flank of the Mid-Atlantic Ridge that's circled or boxed in red here. And this is a sort of zooming in of that site. It's on, like I said, 8 million year old crust. And the reason that this site was chosen is that it's actually a really well characterized site. Like back in the 1970s, when seafloor spreading and plate tectonics was still kind of a controversial issue, they drilled the ODP at that point, drilled into the crust, just looking for good rocky basalts. Originally, and they picked this crust, or they picked this site because it did have sediment. And so if you're drilling into the sediment for 100 meters or so, that stabilizes the drill, and then it can go into the basalt underneath it. So they drilled down, they got their basalt, they took their core out of out of North Pond, and to their surprise, what they found was that open borehole now was sucking seawater down into it. A lot of seawater, like 1,000 liters per hour seawater. And it actually did that for about 20 years until there was another IODP leg that went to CORK in the late 90s. So there's been a lot of work that's been done to sort of understand the hydrogeology of the site. It was drilled and corked again with these sort of modern facilities that allow for you to sample fluids with low contamination in November 2011. And those two sites are the yellow stars here, 1382A, and this one's close to where that original hole was drilled, the one that sucked seawater, and then this other site up here, 1383C. So that was drilled, those holes were drilled, they were cased, they were instrumented and plugged. And then there were cruises that went back to sample the fluids in the spring of 2012. So that was less than a year after the original holes were drilled, 2014, and also one in 2017. And that was this IODP Expedition 336, the one that went to drill these holes. So here is a sort of, here's a picture from Jason of what the wellhead looks like. So the top of the borehole is instrumented, and it has this platform where you can put sort of a pumping station. And coming out of here, you can see that there are 3 lines, and these 3 lines go down to different depths through the sediment and down into the crust where there's mesh, so it can pull up fluids from particular depths. So you go here with an ROV, you set this sort of microbe sled thing down, you hook up the fluid lines to the pump, and you fill Tedlar bags. That's how you get your fluids. Okay, so what do we know about this North Pond site, right? Like here's a a map of the site again, you can see that it's kind of surrounded by these rocky outcrops. After it was noticed that there was a lot of seawater kind of being sucked into the crust, and like, you know, what was that all about? There was repeat expeditions that were done there to sort of measure heat flow, and then additional ones that were done just before that IODP leg. So this is a summary figure that shows those heat flow measurements, and they're color coded such that the gray and the blue are kind of cooler, less heat flow, and the red and the yellow and the orange, that's more heat flow. So this is that the site that was originally drilled. You can see that it's gray and black, and there's not very much heat flow here. You can kind of understand this because if you can imagine that if there's seawater, cold seawater being delivered down into the sediment, it's going to, or into the rocky crust, it's going to cool that area off. So we have little heat flow here where that recharging sediment, recharging fluid is going into the crust. And then in other locations in North Pond, there's more heat flow, and you can imagine, so here and then here, you can imagine that in these locations where there's more heat flow, it's likely that there's less connectivity to seawater. The fluids that are probably down under those locations have been isolated from the bottom ocean for longer periods of time. So the original plan was actually to put in 3 of these observatories. There was one that was supposed to be here, but that one never, it was not a successful attempt to drill that one and have it corked. So using these data, these heat flow data, there was a sort of model created for what the flow must look like underneath North Pond. So this is kind of a cross section of North Pond going here from southeast to northwest. So from roughly this area kind of to this area. And this is the sediment here, and this blue arrow represents the water that's going, that's, you know, infiltrating down through this hole. And there are values here that I'm not sure if you can read, but here's 5 degrees here. So it's expected, it's modeled that near that first hole, there's 5-degree fluids, and the fluids warm up to a maximum of about 25 degrees based on this model before they're coming back out, probably over here somewhere. So unfortunately, that's still the best understanding that we have of the temperature regime underneath North Pond, because there were some instrumented or sort of temperature loggers that were put down in strings in these holes after they were drilled. And we only have 2 of these holes, right? This is the second hole. And in that warmer, more northern hole, those temperature loggers are now in the bottom of the hole. So we're never going to get those data. But we think that this is a reasonably good representation of what the temperature looks like in the crust beneath North Pond. Okay, so before I start showing you what the data tell me, I'm going to, so let's zoom out a little bit and think about how these hydrothermal vents and hydrothermal fluids in general connect to larger scale sort of ocean chemistry, ocean biogeochemistry. So it's pretty well established, I think, that inorganic, that hydrothermal vents are important for sort of inorganic carbon cycling. There's a lot of iron that comes out of these vents. As I said, there's magnesium that is removed in the vents. And this is, I think, pretty well understood. The carbon implications, though, the contributions of hydrothermal fluids to carbon cycling is not thought to be very significant, because if all you're looking for is the kind of carbon cycling that happens along those sort of ridge axes, it's not a large enough flux of fluid to make a big impact on the carbon cycle. It's also pretty poorly constrained. So here's the carbon cycle. You've probably seen this figure before. You may have used this figure before. It shows the reservoirs of carbon, organic carbon and inorganic carbon, and the sort of major fluxes between them. And you can see from this figure that there's huge amounts of organic carbon in the sediments and in sedimentary rocks, and you don't see any crust underneath here. And I'm not going to argue that that's wrong. From what we found, there's very little carbon in the crust. But what I did want to highlight in this figure is that there's this POC reservoir, not POC, sorry, the DOC reservoir, that is a pretty large reservoir of organic carbon It's also one that's not really well understood. It still has its share of mysteries. It has a wide range in its residence time. And to sort of understand organic carbon cycling in the hydrothermal fluids and connect it to larger scale ocean organic carbon cycling, I'm going to use sort of the DOC reservoir and radiocarbon. All right, so I'm sure that most people in your department don't need this kind of introduction to radiocarbon, but I'm going to use radiocarbon because it's a great both clock and tracer for sources in the marine environment, right? So it's produced in the upper atmosphere by the interaction of high energy neutrons with N14, which turns into C14 and gets incorporated into CO2, and it mixes quickly throughout the atmosphere and is taken up into terrestrial by terrestrial plants. It mixes a little more slowly into the surface ocean, and that creates a useful offset that we call the reservoir age. And then it mixes even more slowly into the deep ocean, and that you can see here, high concentrations of C14, low concentrations of C14. Hold on. Okay, so this makes it, because of all of the contrasts in different reservoirs, it makes it a useful tracer of where your carbon is coming from in the marine environment. And I'm going to use it as that. I'm also going to use it kind of as a chronometer. So, and also I should point out that I'm going to use delta C14 units to describe my radiocarbon values. Delta C14 goes from 0, which is the amount of C14 that was in the atmosphere in 1950, to minus 1,000 per mil, which is undetectable by our best methods, and that's about it's about 55,000 years old. 55,000 years old, yes. And there's also some positive values that were produced by bomb testing, by nuclear bomb testing. Okay, so back to DOC. So the thing about DOC that kind of makes it mysterious is it is old. Right, it has a really old radiocarbon age, particularly in the deep ocean. It has a radiocarbon age of somewhere around 5,000 years, which is equivalent to somewhere around 5 ocean overturnings. Right, and when the concentrations and delta C14 values of DOC were first reported, it was noticed that they both kind of followed the same profile, where they're high concentrations and C14 enriched DOC near the surface ocean. So here's the data plotted on a log scale. And once you get down to about 1,000 meters depth, there are lower concentrations of DOC and constant down to the bottom, the water column, and also lower delta C14 values that are kind of constant down to the bottom of the water column. And that's consistent with this two-component model of rapidly cycling or labile DOC and refractory DOC that sticks around for a long time. So this is evolved over time to more sort of subdivisions of these labile and refractory components, but we still kind of divide organic carbon, dissolved organic carbon, into these reservoirs. This is a sort of more recent version of the division into labile and refractory pools. Here's the original a DOC cycle, and then in this revised version, you split deep ocean DOC into a really refractory component. So not all in this model, not all deep ocean DOC is refractory, but there's the really refractory stuff, and then there's a more labile fraction that's less than half of it, now less than half of the concentration, that comes from hydrolysis and dissolution from sinking particles. And the other thing that I wanted to point out about this figure is that now you see hydrothermal DOC that's kind of included. It's lumped in with sedimentary DOC and methane seeps, and there are no values associated with these, but it is included now in this sort of DOC cycle. All right, so this is an interesting question also because it's a little bit of a puzzle. If you are a bacteria and you live in the deep ocean, your job is to kind of look for energy and for food, right? And down there in the oxygenated deep ocean, DOC is food, but it's really not bioavailable. I guess it doesn't get eaten. And we don't fully understand why. There is one hypothesis that's been put out there that maybe there's too many different molecular, like the DOC is too heterogeneous, and it's also very dilute. So maybe it's not worth it for bacteria to try to access this carbon. It's too energetically unfavorable to even to sort of come up with strategies for dealing with this heterogeneous carbon. I'm going to come back to that at the end of this talk. Okay, so now back to the Mid-Atlantic Ridge site. So this site is called North Pond. The sites that I'm going to show you data from are these two that are starred. I'm going to give you like a schematic cross section of what the sites that were installed in North Pond look like. So there's 4 corks that are installed there now. This is just if you took the figure, the North Pond figure, and you flipped it down. This is what it would look like in cross section with the sediment sort of in the darker or in the lighter brown and the basalt in the darker brown. This is that hole 395A, the original one that was drilled in 1975 and that sucked seawater down. It's been corked. The hole that was drilled nearby that is drilled through about 90 meters of sediment, and it has only one depth in the basalt where you can recover fluid from. And this is the sort of cooler site where we expect recharging seawater to be pretty close, pretty proximal to where we're sampling fluids. And then the other site, the one that was farther north, that's circled in red, that's where we expect the fluids to be a little bit warmer. It's installed in a shorter sediment column, only 40 meters. And this one goes deeper down into the basalt, and there's 3 sections of mesh, so you can sample fluids from 3 separate depths within this cork, within the borehole. Okay, so I'm going to show you now the very first data that came up here, and some of these data I would not use again, but it's worth showing, I think, because, well, first of all, this is kind of what it looks like when there's the sled on there and the fluid lines attached. A couple of other views. So the expectation was that the fluids that were going to be recovered from North Pond were going to look like every other kind of hydrothermal fluid that anyone had really ever seen before, right? It was going to be, if not anoxic, but at least low oxygen. They expected to, I'm saying they because I wasn't involved yet at this point, but they expected to measure methane, they expected to measure hydrogen concentrations, ammonium, and they expected fluids that were a little bit warmer. So this is, in this table, there's bottom seawater on the top, and then this is the site, that cooler site, and 3 depths at the warmer site. So this is not very warm, right? The bottom seawater temperature is only about 1 degree or 2 different from the fluids that are coming up. So there is a big asterisk on all of these temperatures, right? We don't really know what the in situ temperatures are because we don't have the temperature logger data. But this is the temperature that was logged on this pumping sled after the fluids had been recovered. So the fluids have been pumped through the sediment at that point. So it's either through 90 meters or 40 meters of sediment, and they probably cooled off, but they're not really far off. These weren't screaming hot fluids that cooled off substantially by the time they got to the temperature sensor. The pH doesn't change very much. The oxygen concentration goes down. That's interesting. The DIC reservoir doesn't seem to change very much. The DOC reservoir, again, I'm not going to use these values any more than this slide, but they do go down, and that's also interesting. And magnesium, if this was, these were fluids that had experienced high temperatures and had reacted with the rocks, they would have been expected to have lower concentrations of magnesium, and they don't really. So there don't seem to be very much high temperature reactions going on. Right, so overall, the fluids look a lot like seawater, maybe except for the oxygen and the DOC concentrations and a few other trace elements. They look very similar to seawater. And then we have this sort of modest oxygen depletion. And one of the explanations for the oxygen depletion is that it's being diffused upwards into the sediment column. So I didn't include this this figure. But if you look at the oxygen concentrations in that whole column of sediment, what you see is a normal sort of high oxygen at the top of the sediment column and then decrease with depth. And once you get down to a certain depth, you see oxygen concentrations increase again because the oxygen in the fluids that are flowing underneath the sediment column are diffusing upwards. So there's some loss of oxygen from that, but that doesn't explain all of this oxygen decrease. But one game we could play is to try to see whether there's any correlation between the DOC concentration, the decreasing DOC concentration, and the dissolved oxygen. So these are not the same dissolved oxygen values that I showed you before. These are remeasured. Dissolved oxygen, or sorry, DOC is really hard to measure and it's really easy to contaminate. So I threw a lot of measurements away because they just seemed contaminated. And these are the best measurements and the most repeatable measurements that that we have, or they're the only ones that are plotted on this plot. So this is a summary of all 3 of the sampling years from 2012 in the open circles, 2014. So this was not long after the drilling, 2014 in the gray circles, and 2017, the most recent sampling in the black circles. And they fall pretty well on that linear line. So it does seem like DOC concentrations and oxygen concentrations are correlated. They seem to decrease together. But you can look at that slope of that line and notice that it doesn't look like Redfield. You know, it looks like a lot less than Redfield. So compared to the amount of DOC that's removed in these fluids, there's extra loss of oxygen. Right? And some of that, again, can be attributed to diffusion upward into the sediment, but there's still excess loss that that isn't really well explained here. It's still not very well explained. So the other place to look for clues, so we could have decreasing DOC because it's being oxidized. We could also have decreasing DOC because it's lost by adsorption to the crustal surfaces, or incorporated by the microbial population into biofilms that are on the crustal surfaces. So one other place to look to see whether that DOC is really being oxidized is the DIC reservoir, the inorganic carbon reservoir. So that DIC reservoir is so much bigger than the DOC reservoir, but the delta C13 values are sensitive to the source of that inorganic carbon, right? So I measured the delta C13 values of all the crustal fluids, and they all turned out to be a little bit lower than what you would find in bottom water, right? So here's the delta C13 value of the fluids, and here's bottom water in 2 years. It's about 1 per mil. It's the same as you would find kind of anywhere in this area of the North Atlantic. And then I have the same sort of color scheme for the samples that were collected in 2012, 2014, and 2017. So in 2012, there are some pretty large sort of depletions in C13. In 2014, less so, and in 2017, even more modest. However, in all cases, the DIC in crustal fluids is more depleted in C13 than it is in bottom water. And the way that I interpret this is that there is some oxidation of organic carbon with a delta C13 value of around 20, we can guess like 22 per mil, that's contributing CO2 or DIC to that total reservoir. So the average, here's the average values again. This sort of emphasizes the fact that in 2017, it seems like there's less oxidation of organic matter, less contribution of that oxidized organic matter to the IC reservoir. So you can do what any good isotope geochemist would do and make a isotopic mass balance and try to determine what fraction of the total DIC reservoir comes from oxidized organic carbon. And when you do that, you end up, so I split that samples, the most recent samples from 2017, the ones that are farthest away from the drilling disturbance that happened in 2011. I split those samples into sort of a different category as the other 2 samples. So the first 2 years that we were able to sample fluid was 2012 and 2014, and in those fluids, this mass balance suggests that 1 to 5% of the DIC comes from oxidized organic carbon. In 2017, farthest away from the drilling disturbance, it's a smaller amount of IC that comes from oxidation of DOC. So this also agrees with that's equivalent to about 5 to 100 plus or minus 40 big error bars of that DOC, of that DIC coming from oxidized organic carbon. And this is a small plot. I realize that, but this is kind of like a squint your eyes kind of thing. You don't really have to look at the numbers. But there were cell counts, microbial cell counts that were done for all of these fluids in all of these years. And again, you can see kind of a lot of the open circles and the gray circles are up here, higher cell density. And the black circles that represent 2017 fluids are again like low cell density. So if there is, if that oxidation of DOC is being done by microbial activity, there are fewer microbes and there's a smaller amount of that DOC oxidation effect in 2017. Okay. So the other thing we can use that DIC reservoir for is to use it kind of as a clock to take a measure of how long these fluids have been isolated in the crust. And I realize I'm taking a lot longer to say this than I expected to, so I'm going to speed up a little bit. But if we do that, if we subtract the radiocarbon age of the DIC in bottom water from the DIC that we're pulling up from the crust, what we get is an estimate of that first site where the seawater is recharging close to the site. We get a radiocarbon estimate of about 200 to 500 years that these fluids have been isolated in the crust. And the warmer site, the one that's farther north, we get more like 2,000 to 3,500 years that these fluids have been isolated in the crust. So if you're familiar with these kinds of aquifer residence time estimates, this is much longer than you would get from other estimates like modeling and heat flow. And that, I think, is reflects the fact that you're getting fluids from these corks that are partially the really efficiently flowing fluid through well-connected pores and cracks, and partially fluids that might be more stagnant and aren't as well represented in like heat flow models. Okay, so here's a sort of visual representation. I put lots of effort into making this figure, and we never published it because it's actually a bad representation of what is actually happening with the fluid. It's not reasonable to expect that you have bottom water that is entrained in the crust and it goes past this first 1382A site and then goes to 1383C, right? There's lots of very complicated flow paths, and the fluids that we pull up from one location, they're not, earlier, or older, or younger than fluids from the other location. They're just different. But there does seem to be kind of a settling into a pre-drilling state after a few years after these were sampled. Okay, so now we have these isolated fluids, and we know that the DOC concentration goes down. But the question is sort of what features in that DOC can tell, what can we tell about the processes that cause that from the features in the DOC. So the first thing one of the things that we did was we collaborated with Torsten Dittmar's lab in Germany, and they measured the molecular spectra of the DOM on one of these high-resolution FT ICRMS instruments. And if you just kind of squint your eyes, so here's just the spectra, right there. It's just the spectra that comes off the instrument, and you can tell just by eyeballing it. Here's bottom water, that 1382A site, the one that's closer, the cooler site, and then 3 depths from 1383C. And here's sediment pore water from the water column, or from the sediment column above these sites. And you can tell that these all look alike, and the sediment pore water looks different, right? So we can kind of assume from this that sediment pore water is not a big contributor to the fluids that are flowing through the crust. You can see that kind of in a large scale way, and also if you zoom into these particular narrow windows of molecular mass, you again see a similarity between bottom water and all the crustal fluids, and the difference between those fluids and pore water. So in all of these cases, I've used kind of the same coloring convention where, and there's a key down here where bottom water is blue, that first site that is cooler is green, and the 3 depths at the warmer site are orange, red, and and purple. I'll just keep kind of going with that. So the other way, besides just eyeballing it to see this, is to incorporate this into a sort of a PCA analysis. And again, seawater and crustal fluids kind of grouped together. And sediment-borne water is in its own sort of part of the plot. So we can be pretty confident here that the origin of the DOM is from bottom seawater. It's a sort of marine DOM. It's not from pore water. It's not from microbial activity in the crust. It's that sort of deep water DOM. Okay, so if we look a little bit deeper into this FTI CRMS data, here again is sort of a summary of the oxygen concentrations and the DOC concentrations, and now I'm plotting everything against incubation time that I am deriving from the DIC, from using the DIC radiocarbon ages as a clock. So both of these, with incubation time, DOC concentrations go down, oxygen concentrations go down. Okay, so now we're looking at the dissimilarity between the samples, and the dissimilarity goes up. Right here you have bottom water. The 1382A site is more similar to bottom water, and these warmer site, the DOM in warmer sites are different, are more different. This is the aromaticity index. Again, you see a change sort of with incubation time. We have bottom water with a low aromaticity index, and you have an increasing aromaticity index with time. And so what could this mean? This could mean that what's left in the DOM reservoir in the crust is preferentially the condensed aromatic structures. So this could maybe be something like black carbon that's preferentially left behind and not consumed by microbes that are in the crust. So this is the CRAM, or it's a carboxyl-rich alicylic molecule. So in other words, cyclic organic carbon with polycyclic structures, but not aromatic structures. And this is less clear here, but you have this trend with time of increasing CRAM percentage. And CRAM is thought of as a refractory component of marine DOM. So that's also what's left behind as microbes are chewing away at this organic this DOM in the crust. And then there's another sort of proxy called the degradation index. A degradation index is made up of just 10 masses that were observed to be correlated with in the open ocean with sort of aging of DOM. And the degradation index also with incubation time goes up. Because this was sort of developed in the open ocean, these are the oldest degradation index values that I've found anywhere. So what's left in the crust seems to be really molecularly degraded. The fraction of DOM that is maybe the least bioavailable and most degraded. Okay, so what can the carbon isotopes tell us? Before I go to the carbon isotopes, here again is a summary of all of the DOC concentration values. They fall pretty linearly on this line with incubation time. So from that, you can calculate a loss rate, which is pretty slow, but over thousands of years, it accumulates to a removal of, say you have 40 micromoles of DOC that's being delivered into the crust with seawater. 20 micromoles is lost by the time you get over here to a few thousand years. Okay, so I'm going to start with delta C13 because we were talking about sort of the molecular preferences for that removal previously. So there's not a clear trend here, but there is an overall sort of decrease in delta C13 value with incubation time in the crust. Not only that, you can see that I've again colored these so that the open circles are 2012, the sort of lighter colors are 2014 fluids, and the darker colors are the ones that were recovered in 2017. And there's a difference between these years. So I would say you kind of don't expect a lot of like dynamic change in an environment like this where the cell concentrations are so low, but things do seem to be changing, and maybe that's more of an indication that you're sampling greater or less fractions of the really efficiently flowing fluid versus the fluid that's kind of stuck in the cul-de-sacs and the dead ends of cracks in the crust in different years. It may not be that the whole reservoir is that dynamic. But there is this selective removal of the more C13 enriched components, right? And so what could that mean? so there you could have enriched C13, or sorry, depleted C13 from methanotrophic organic carbon, but we don't see any methane, so we're going to just cross that out. Terrestrial organic carbon could be depleted in C13. So I mentioned on the previous slide that the explanation for the increasing aromaticity index could be that what's left sort of in the remnant DOC is black carbon. So if that black carbon was produced in a terrestrial setting from terrestrial fires, that could be an explanation for the decreasing delta C13 value of what's left, that DOC that's left. The other thing that I think is really interesting as a possibility is that if that CRAM material, you also have that increasing CRAM index, right? So down here at the bottom is what CRAM could look like. And it has a really similar structure to the hopanoid. So lipid biosynthesis pathways have, and the end result is that they have they produce molecules that are more depleted in C13 than most of the other biomolecules that you would find in a cell. So maybe what this is telling us is that if CRAM, that CRAM could have an original source from lipids, and they may also then carry a depleted C13 signature. Okay, so radiocarbon. Here again is the DOC concentration, same as in the previous slides. Here's the delta C14 value. So the delta C14 value goes down, and this could be for multiple reasons. And the other thing I wanted to point out is, as you start to look at these figures more, you kind of see, or at least I see, that there might be a different rate of removal of DOC kind of in the first in that first section between bottom water entrainment in the crust and that first cooler location, and the thousands of years it takes to get to the second location. So if you were to separate the DOC loss into an initial rate, it's much faster. It's 0.02 micromoles per year compared to the overall. But this sort of pattern could be caused by multiple things. It could be caused by radiocarbon decay. Hold on a second. Radiocarbon decay. Or it could be caused by selective removal of DOC that is enriched in C14, or by the addition of DOC that has a particular delta C14 value, either through chemosynthesis or sedimentary DOC diffusion. So one thing you could do is you could take that age that we got from the DIC reservoir, and you could use it to correct this delta C14 value. So if I do that, you see this difference between that kind of initial removal of DOC and the removal of DOC that happens over thousands of years. You see this much more clearly. So if radiocarbon decay was the only explanation for the aging of the DOC, all of my points would be up here at 0. But instead, they're all kind of down here around -500 per mil. And what that says to me is that early on, when the seawater is entrained in the crust, all of the really young C14 enriched DOC is removed. And then what you're left with by a few hundred years and through the rest of the sampling time, the time that we can see from these samples, the few thousand years, there's not much change after that in the radiocarbon concentration of the DOC that's left. Right, so here is my list of influences, right? And we're crossing out radiocarbon decay by correcting for it, and we've already kind of crossed out sedimentary DOC diffusion. So what we're left with is this selective removal of DOC, isotopically selective removal of DOC, and potential addition. So the last point that I wanted to make about this is that whatever that removal mechanism is, it seems that the C13 preference and the C14 preference are tied. So what I have plotted here, this is from the paper, so it doesn't include 2017 fluids, but it's the delta C13 value plotted with incubation time, right? And there's not much of a relationship here. But if you take the delta C13 value and you plot it with the radiocarbon age instead of the incubation time, sort of another measure of time, you do get this relationship. So whatever the removal mechanism is that's getting rid of the DOM that's in the crust, the preference is for organic carbon that is both C13 enriched and C14 enriched, right? So we get this this removal of DOC initially that's Now this would have been better to put on another slide. But it's a faster rate. It's enriched in C14. It's less molecularly degraded. It has less % cram and less aromatic. That's what's preferentially removed. So what this kind of supports was this this paper that I had included earlier, that maybe there is that refractory deep ocean DOC has a component in it that is more bioavailable, younger, and less molecularly degraded, and that's what's removed first. And then once you get a few hundred years into that incubation time into the crust, what the microbes have to deal with is just that really old molecularly degraded DOC. Okay, here's the pretty picture that summarizes all of that. Right, here's deep ocean DOC with about 40 micromolar and a radiocarbon age of about 4,700 years. It's incorporated into the crust, and it's removed kind of in two phases. That initial removal of the sort of more C14 enriched and less degraded DOC, and that removes about 7 to 10 micromoles of DOC, so less than half of it. And then you have this sort of phase 2 of this removal of about another 10 micromoles of DOC over this few thousand-year time scale, and the average age of that removed DOC is about 3,200 years. Right, so these are microbes that are eating that organic carbon, that DOC with an old radiocarbon age, that the bacteria in the deep ocean don't want to eat. So that's a summary of what I kind of walked through. Like if this is DOC and the concentration of DOC with depth in the ocean, you lose the sort of really labile stuff, and then you have this deep ocean DOC that's refractory, but split into a less refractory and a more refractory section. Okay, so can we extrapolate this into like a globally relevant number? The total DOC removal rate, refractory DOC removal rate in the ocean is here 3.1 times 10 to the 12th moles per year. Some of that can be explained by adsorption in the open ocean and gel formation, and that's 2 times 10 to the 12th moles per year. So that leaves about 1 times 10 to the 12th moles per year of a missing DOC sink. So hydrothermal circulation on ridge axes and on warm off-axis circulation can't explain this. Right? It's order of magnitudes less. But if we take our values and we sort of extrapolate it to off-axis, older than 5 million-year-old crust, what we end up with is an order of magnitude less than this hole here, but a pretty significant fraction of it, perhaps. We got about 1 times 10 to the 11th moles per year, or maybe 10% of global deep ocean DOC removal by these hydrothermal fluids. Okay, so I'm about to run out of time, but I'm going to tell you one other thing that I think is interesting. You may have noticed that the purple dots are kind of don't fall in the pattern of some of the data that I've showed you. And we thought initially, when we only had 2012 and 2014 data, that maybe this meant that there was some chemosynthetic DOC that was being contributed at the very deepest depth. It seems like it's not as depleted in C14, it's not as depleted in C13, it's kind of less degraded, less cram, less aromaticity index. So we wrote a proposal about this, and that's why we went back in 2017. And the answer, we think, is that there's not chemoautotrophy. So this is data from a colleague of mine. And what she did was she incubated the fluids. Right? If you look at the geochemistry of the fluids, there's no sort of redox potential there except for oxidation of the DOC. So she took lots of replicates of these incubations, and she incubated for at 4 degrees C and 20 degrees C, bracketing the temperatures we expected to see. And she added labeled acetate, bicarbonate, and methylamine. So maybe they need to take up organic nitrogen. And she found lots of uptake. These are bulk rates of acetate. She also found surprisingly high uptake of bicarbonate, and particularly in these warm, middle, and deep fluids, and not very much uptake of methylamine. So she did this also on a single cell level using nanosims. Right, and these are it's the summary of her nanosims data. And again, here's bicarbonate here. There's lots of bicarbonate incorporation if you look at it by single cell, and acetate is these A. So the acetate incorporation rates, sort of envelope of probability, and the bicarbonate incorporation rates are similar at 20 degrees C, and they're also similar, A for acetate, B for bicarbonate, at 4 degrees C. So what we think is happening here, she didn't add any energy to these incubations. What we think is happening is that it's anabolism, that these microbes have decided that the organic carbon may be refractory and hard to deal with, but they're perfectly fine as an energy source. So they could be just oxidizing that DOC as an energy source, and then incorporating DIC using kind of anapolaritic pathways and doing that production of fresh organic carbon because the DOC makes poor metabolic intermediates, so they need to make brand new ones. Okay, so my quick lessons. Lots of DOC is removed by microbes in cool hydrothermal fluids, and it's returned, the DOC that gets returned to the deep ocean is depleted in C14, C13, and it's degraded. That hypothesis that was kind of put out there that dilute concentrations of DOC is really what limits the bioavailability of deep ocean DOC. We are seeing oxidation of DOC from 40 micromolar down to 20 micromolar. So it does not seem that dilute concentrations are a real limit here. It also seems that from those incubation data that I showed you that the bacteria are likely employing sort of unique strategies to access refractory DOC. They're not incorporating it like a normal heterotroph would, potentially. And these isolated fluids, what's left in that fraction, that half of DOC that's left that we recovered from that warm site is probably the most refractory fraction of DOC, that's hidden in deep ocean DOC, but it's kind of left naked in these fluids. And the last thing is that when you normally think about fluids that are flowing through the crust, the methods that are used to access the most efficiently flowing fluids. And by pulling them up and looking at them sort of in detail, at the organic carbon in detail, we're getting a bigger picture of an integrated picture of the sort of efficiently flowing fluids and the less efficiently flowing longer incubation time fluids. It's a better picture of total biogeochemistry. And those are my take-home lessons. I'm happy to take any questions.
All right, thank you very much, Suni. That was very, very entertaining to see what's happening in this unique environment. So, as is normal, we want to try to open up questions first to any students that may have questions. Anybody have any questions from the student body?
Okay, I'm just gonna jump in here. This was so cool. Thank you for this talk. Were you surprised to find a preference for isotopically enriched material? I guess my expectation energetically would be that something would prefer to take up something lighter. Does that make sense?
Yeah, well, that would be true if it was kind of all the same thing, all the same molecule. So I think what drives the preference has got to be something related to the molecular structure. I think there's maybe some fractions of the DOM that are intrinsically harder to access than other fractions. So I think you have to look more specifically into the molecular distribution to answer that sort of preference question.
Okay, cool. Thanks.
Any other student questions? My lab group was chatting on a different channel about this talk quite a bit. And I know that the students in the chemical oceanography class have had their fill of mass balance equations, or what I usually call mixing models. So I was happy to see one of those in the talk.
Yeah, how can you not do that?
Right. All right. I guess we can open up questions to everybody then. Anybody else with any questions for Suni? I
have a question.
Yeah, go ahead, Dave.
The student part is over, correct?
Yes, go for it.
I just wanted to, I know this isn't your expertise, but it was very fascinating to me. I had never seen this plot that you showed, that the fluid pore waters would kind of come to a termination in motion after 65 million years. And it was interesting to me because the Parsons and Slater's subsidence curve showed that the ocean lithosphere flattens out after 80 million years. And so I'm just curious, I know this is just hypothetical, but do you think that that might play a role in what's causing the lithosphere to flatten out to some degree, with the exchange of heat now becoming purely more conductive than the flow of water through the lithosphere?
I think that is a, I don't know if I can answer that one. I don't, yeah, so the flattening out of that lithosphere curve is more than, that's way beyond my expertise.
No, but it's just an interesting observation, and I'm glad you showed that figure. I thought that was very interesting. Thank you.
I wanted to ask a question about that same series of figures in the beginning of the talk, because that was surprising to me as somebody who does some research on river to ocean carbon cycling. You compared the overall flux to rivers and oceans, and it was basically on the same order of magnitude from the hydrothermal fluxes, and that really surprised me. But you also brought up the Arctic Ocean and I realize it would be hard to do this work in the Arctic Ocean, maybe at least for the next few years, and then the ice will be gone and we could do it. But it's sort of an enclosed ocean or semi-enclosed ocean with quite a bit of rifting, even though it's pretty slow rifting. But I wonder if some parts of the ocean might have an even stronger signal from hydrothermal exchange, like the Arctic Ocean, because you basically have a smaller volume and less circulation through it compared to the hydrothermal fluxes.
That would be really fun to try to think of where to go so this is a totally different kind of location, and I just, the enclosed basin part is what makes me bring this up. But the Guaymas Basin, for example, is really, really narrow.
Okay.
And there's tons of organic matter in Guaymas Basin, so you couldn't do the same kind of thing. But you can see in the DIC, I think Patrick Rafter has shown that you can see that the DIC is older than you would expect it to be because of the flux of old hydrothermal carbon that's being released. So it's possible. But the other thing about the Arctic Ocean that makes it complicated, I mostly know about the other end of the Arctic Ocean, I guess, the older basins, but there's so much resuspension and redistribution of sediments that if you got old values, it would be hard to be able to separate those two.
Yeah, and I know, for instance, in the surface of the Arctic Ocean, because of DOC from Arctic rivers increasing as Siberia warms up, that's actually causing the DOC to become older in the Arctic Ocean. So it might be, there might be competing influence, even though it's a little bit more stagnant than the open oceans. But you bring up an important point too, that when we're talking about these deep water masses, you really have to look at the bathymetry. There's some sort of semi-enclosed basins in the middle of the open ocean, because you're really only looking at deep water exchanges here. So yeah, thank you.
Yeah, but that also so that makes that collaboration that we had to get the FTI CRMS data really valuable, because we also had too many explanations for the values that we had until we could start crossing some things off the list just based on non-isotopic data, other data. Right.
Yeah, good point. All right, any other questions out there? All right, so let's open up our mics and give Suni a thankful round of applause for her talk today. Well, thank you very much. It was very entertaining.
Yeah, thank you. Thanks for inviting me. I'll see you guys.
Yeah, it was wonderful to have you.
Yeah, it's good to see you again.